All right, good evening. So for today's video note topic is under karyotypes. And our essential question is, can you define karyotype and how chromosomes are arranged? So first of all, we need to remember that in human chromosomes, we have sex chromosomes to determine if we're male or female. And so 2 of those 46 is what actually determines our gender. So females, you would have 2 X chromosomes, and a male, you would have an X and a Y chromosome. And here's a picture of the difference in size.
So chromosomes, what are you looking at? Essentially, they're groupings of genes. And so when you see bands on chromosomes, you can stain them like we did our cheek cells or something like that earlier in the year. There's banding on there, and that shows you the different genes. And just as a sort of side note, there's 1,200 genes on the X chromosome, whereas the Y chromosome is a little over 10% the size of an X chromosome at 140 genes only. The other 44 chromosomes, other than the sex chromosomes, are referred to as the autosomal chromosomes, and they account for all kinds of traits.
Okay, so if you look at a karyotype is a picture showing you your full complete set of chromosomes, and we call them the diploid set. Remember, in all of your somatic cells, you would see this could be laid out for you from all of those body cells. And so you'll have one chromosome from your mother and one chromosome for your father, so you'd have 23 pairs for a total of 46. And you notice that they are numbered here in this karyotype, and here is your X and Y chromosomes right there. All right, so karyotypes are arranged by 3 things, and the first one is size. So the size of the chromosomes themselves. So when you lay them out and you see a picture of a karyotype, they're going to go from the biggest or the longest to all the way to the smallest or shortest, and then the X and Y chromosomes go at the end.
Okay, another way that you look at arranging chromosomes has to do with where the centromere is located. So if you want to think of above or below the line of where the centromere is, as you can see here labeled P or Q, they basically end up in different spots, roughly in the middle, metacentric, towards the top, sub-metacentric, or really almost completely at the top, called acrocentric.
And then finally, the third part is the genes. And so these light and dark bands are the genes, and so there is a pattern, and so you would line up your mother's chromosome to its father's homologous chromosome, and you line them up, and that's another way of the karyotype being set up.
All right, how do you use a karyotype? Well, first you have to obtain a tissue sample. Usually this is going to be done actually in utero, right? If you want to know a potential condition that your child might have, if there's something that runs in your family, you can get a sample from a lot of different places, but you essentially need to grab a somatic cell and take a look at it.
Once you have the cell, then the scientists are going to allow for that cell to go through mitosis until it gets to metaphase. And once it's in metaphase, then the chromosomes are all lined up in the middle.
This would be a point you would add essentially hit the pause button or something called colchicine, which is just a chemical that essentially stops the chromosomes in place. And then that allows you to blast the cell open and take a look at them.
Which is important during metaphase because the chromosomes are condensed. You can actually see them in the chromosome form. You can see that they're sister chromatids, and you'll be able to look at a homologous mother and father set pair.
So as I said earlier, you blow the cell up, you rupture the cell to extract the chromosomes, and of course they're not just going to line up, they're going to be kind of a mess. You take a photograph, and then you can arrange the photograph based on those criteria we talked about earlier, the size and where the centromere is located and the banding patterns.
And then once you've arranged them, scientists will put together the karyotype photograph shown here, and then they're able to analyze and determine whether there are any conditions or anything that they want to be aware of.
So why do you do these? Well, they can tell you certain things for sure, the sex of the child, those types of things. But they can also tell you if there could be potential problems. And one of those problems, most of the problems arise from something called non-disjunction. And what that is during meiosis, sometimes homologous chromosomes don't separate. And what that can lead to is either too many chromosomes or not enough chromosomes.
So an example of non-disjunction could be something called trisomy 21, and tri is a root word meaning three, and so what happens is you get three copies of the chromosome number 21. So here is a karyotype showing on number 21 three chromosomes when there was only supposed to be two, and this is something known as Down syndrome.
Another one is another trisomy, in which a non-disjunction leads to an extra chromosomes, Patau syndrome, and in this case, you have three copies at chromosome 13.
Another example is trisomy X, and you're getting this tri, meaning they're getting another extra copy. And so this would be when females have three X chromosomes instead of two on their karyotype.
Occasionally, you get something called a monosomy, and again, this is coming from non-disjunction, but in this case, you actually get one fewer chromosomes. So one condition called Turner syndrome has only one X chromosome, but then there's no extra sex chromosome. There's not an extra X. There's not a Y. It's just a single X.